All right, good morning. We'll get started. Today we're covering the connective tissues. Andrew, do you have any announcements about Beacon?
Same place as it was last week. Library, second floor by the elevator, right after this class.
All right, great. The connective tissues are a very broad class of tissues. It's hard to kind of like, in a nutshell, put them all and what they do. They have a variety of functions. And, the thing to start off with is when you studied the epithelial tissues, you really looked at the cells and how they're arranged and how they're layered. But in connective tissues, you barely notice cells. Usually you see fibers all over the place. So, what's more mentionable about connective tissues is the extracellular matrix. It's just, an extracellular matrix. And when we say extracellular matrix, it's those two things. It's the ground substance and the fibers. You don't really see ground substance in a microscope slide, but a lot of water, a lot of protein. Okay. In the ground substance, it's like a filler. Fibers you do notice, a lot of different kinds of fibers. Fibers are what make connective tissue, as the word said. It connects things together, it binds things together, and when you look at it under the scope, you can really appreciate that. The little inset I have there, the GAGs, that stands for glycosaminoglycans. It's like little bottle brush proteins. That's the GAGs. Glycosaminoglycans. Little bottle brush proteins. What those do is they create an osmotic pressure to draw water into the tissue that gives a lot of the connective tissues its buoyant property and makes it a good filler tissue. There's a lot of different fiber types, but what you have to know is it's the cells that produce the fibers. So they give you a few cell types here. Really, I'll just note for now the fibroblast. Fibers are secreted by the fibroblast cells, and cells of any tissue are the life of the tissue. If the cells are doing okay, the tissues are going to do okay. The cells that live in your tissues keep the tissues functional. Now, there's a lot of different types of connective tissue subcategories, and this table in your book kind of outlines it for you, so let me go through that. Subclasses. So there's a big category called connective tissue proper. That top category. What it's saying there is there's basically loose and dense tissues. So under that, you have loose. I'll say CT, short for connective tissue, or dense. So There's a few different kind of loose ones we'll talk about. Well, basically, the cells and fibers, they're not packed tightly together in loose tissue. When you look at the tissue under the microscope, you see more light shining through the spaces between cells and fibers. Cells and fibers. Packed loosely, just to speak simply about it. That's why they're called loose. Now the dense ones, the fibers are packed so tightly together, you have to turn like the microscope light up a little more just to kind of see through it. And there's two varieties of dense connective tissues. You can either be dense regular or dense irregular. In a dense regular connective tissue, all the fibers run more or less in the same direction. And so, like for example, in a ligament or a tendon, where all the fibers run in one direction. Ligament, tendon. Those are good examples of connective tissue. A ligament will connect a bone to a bone, and a tendon will connect muscle into bone. And you can resist tensile forces if you pull in this direction. Resist tension. Tension is a pulling force. So if you pull in this direction, you can resist that. However, this tissue shears easily. I mean, for example, if you were to pull in this orientation, I mean, you'd rip the fibers apart, okay? But I mean, usually the way they're arranged in the body, they're meant to pull in one direction, but they can shear. The dense irregular connective tissues the fibers run every which way. They run this way, this way. There's no, when you look at it, there's no rhyme or reason to it. So you could pull in any direction. So what they usually say is these tissues, they can resist shearing forces. Resist shearing. So we'll look at a few examples of both dense and regular and irregular. Okay, then you have the cartilages. Okay, we'll look at that. They resist compression very well. And the main cell type to notice when we get to cartilages is the chondrocyte. Cyte means cell. Chondro means cartilage. Cell. Chondro means cartilage. If you can recognize the chondrocyte, you'll know you're looking at one of the three types of cartilages we're going to look at. Okay, so just note that for now. So when we get to it, you'll look for it. Bone. Bone's a pretty easy tissue to identify. Another category. Bone and blood. It's known for its marked rigidity. It's a mineralized tissue. And what I want you to notice about bone, you can even see it from the little picture they got there. This inorganic mineralized matrix is many layers. Bone is lamellar. many layers. That's what I want you to see about bone. Lamellar, that word means layers, like peeling off layers of an onion. Many layers of this mineralized bone. And when we look at blood, blood is our only liquid tissue. Blood. Now, if I want to be really technical, they refer to blood as a colloid suspension, or colloid. It's kind of like I always think of Italian salad dressing, where you got the vinegar and the oil, and if you just let the dressing sit, don't the layers separate? Your blood's like that too. There's plasma and blood cells in it. You can just let the blood cells settle on the bottom, but usually a good blood draw, like it's nice and uniform, homogeneous, and that's how blood circulates. It's the blood cells well mixed with plasma. Blood cells, and we don't call them blood cells. We call them formed elements, but that's kind of a weird, archaic term that doesn't make a lot of sense now. Well, just think of them as the blood cell. It's the red part of your blood, and it's suspended, mixed up with the plasma. Blood cells suspended in plasma. And when you look at a drop of blood, you don't see the plasma, you just see these formed elements. And I'll just identify a couple of them for you. You should be able to tell this cell from that cell. So that's all you notice for cells of the blood. You're either a red blood cell or a white blood cell, and I'll tell you how to tell the difference. All right, so that's kind of an overview of all the tissues I want to cover today. And I'll start with the loose. These are all considered loose connective tissues. And as we go through them, you'll see the organs that they come from, and they'll kind of give you a clue as to their function. Areolar loose CTs. Areolar connective tissue is probably one of the more prettier tissues we have. You see this tissue everywhere. Let me see where they describe it. First of all, how do you identify it? Recognize two fiber types, collagen and elastic fibers. I think that's the hallmark of areolar tissue. Collagen fibers. They're the pink ones on the picture. The elastic fibers, they're thinner, but they're much darker, these like dark hairs. And let me get my pointer so I can point to things on the slide. And they point to it too. Any of the pink strands is a collagen fiber, and any of those like dark strands is an elastic fiber. And if you see any dark cell-looking structure, it's going to be the nucleus of a fibroblast cell. You should be able to recognize that too. Areolar. So that's kind of how you identify it. It's pretty much everywhere. They point to like it underlying an epithelium. I described it in that lecture as a basement membrane. saying that they're located everywhere. It really is everywhere. Example, one place, basement membrane. On the slide, they call the basement membrane a lamina propria. That's another name I see used for basement membrane. The function, I kind of like what they put. It wraps, cushions, organs, and it's macrophages, phagocytized bacteria. So it does play an important role in inflammation. So it holds and conveys a lot of tissue fluid. Let's see here. So I'll say wraps, cushions, binds structures together.
That is the function of the
Yeah, that's the function. That's pretty much what it's for. Usually when I dissect cadavers, I mean, I look at it, it looks pretty, but it's kind of I have to like separate everything, and what holds it together are these connective tissues, especially areolar. So when I try to separate layers when I dissect, that's usually how I notice it. Everything's held together so nicely because of areolar tissue. Now here's a picture from our collection. The textbook picture looked much better. But what I do is, as you well know, like I did the last time, I'll do it again today. I kind of have some slides ready. We'll take a look at our collection. But I always include the pictures I took last year. So this is last year's picture. It's decent. With the lights off, you can see something. And what if I point to this dark line here? What would you identify it as? An elastic fiber. The pink ones are harder to point to. They're kind of blurry. If I point to any of these black smudges, what do you think I'm going for there? Yeah, you just put fibroblasts. And I might just say, identify tissue. Areola. The next loose tissue is the only one where all you see are the cells. No fibers. And it's adipose. Adipose tissue, just look for the adipocytes. The main thing to look for. So what you're seeing there is, you can see the cell membrane, and you kind of notice the nucleus is kind of pushed to the side. And the tissue stain is very clear because the cholesterol and lipids that are in there don't take up a stain. Lipids don't stain well. They don't stain at all. That's why it's all clear. So I kind of call this tissue like the tiny bubbles. So if you know what you're looking for, you're just looking for these very faint cell membranes, and looks like tiny bubbles. So there's the picture there. And it's hard to get in focus because you're just trying to get this thin membrane of the cells in focus there. And that's all you see, just adipocytes, nothing else. The function nutrition, of course, because that's your lipid store, but also in the subcutaneous tissue, that's one of its major locations, subcutaneous tissue. regulates body temp. So the more fat you have, the millimeters of fat, you have warmth and insulation. So adipose usually isn't a problem. Usually we try to get rid of it, right? Try to reduce our body fat. But in survival situations, this actually becomes really important. Like for example, if you're trapped in, abandoned in open water at sea, you can't survive very long. But if you're not too far from shore and you're a good swimmer and you have enough millimeters of fat, maybe you can make it without dying of hypothermia. So those are the kinds of questions that you start to consider this as a serious thing for survival, okay? Usually for cosmetic reasons, we like to minimize our adiposity. All right, so I want to move on to the next tissue, reticular tissue. Reticular tissue. This word comes up a lot in anatomy. It means dense network. This tissue has kind of a mesh or filtration function. So let's consider the organs where it's located. I would say primarily spleen and lymph node. Those are the main ones to know. So these are immune filtering organs. How it works is the spleen will filter your blood for pathogens. Lymph nodes filter lymph. Lymph is a fluid, and these things circulate. And so with this mesh-like tissue, do you see the black fibers on there? Notice the black reticular fibers. This thing really dark. It's the main thing to look for when you try to identify it. Look for black stained reticular fibers. And within those fibers, you have all of the white blood cells, the lymphocytes that fight infection. So I'll say WBCs. They live in this reticular fiber net. So when you force fluids through it, you're really filtering all the blood and lymph, looking for pathogens that the white blood cells can react to. Okay, so it really does, this picture doesn't really make it look like a mesh network function, but this picture does that I took. So I'll tropicalize just a little bit so you can see the cells a little better. So all the little cells that you see are being filtered through this mesh net. And the goal is, if there's an infection, if there's a blood-borne pathogen or something in your lymph, that this reticular tissue will filter it out or initiate an immune response. Okay. All right, we're moving on to the dense tissues. Dense regular connective tissue, I would say tendon and ligament are the commonest examples. Just look for the collagen fibers to be more or less packed tightly together and running in one orientation. And it's kind of easy to see from the picture. They're kind of more or less running horizontally across the picture there. You may see the nuclei of the fibroblasts, but they're few and far between. I'll put fibroblasts as well. Like I said earlier, this kind of tissue, the function is resist tensile forces, stresses, resist tension. Here's a picture from our collection. Looks like a plank of wood. But again, they're all running in one orientation. And yeah, more or less up and down. And that's what the tissue looks like. The irregular one, the fibers run every which way. And you know, I don't even know if I like this picture that they took. I always included my own. The example I'll give you for dense, irregular connective tissue is from the dermis of skin. And it resists shearing forces. I haven't taught you skin yet, but the dermis is the underlying layer under the epidermis, which is the very top layer of your skin. Okay, but I'll talk about that more on Monday. There's another picture. This is a 100x, and when you look at a 400x, you can see there's no normal arrangement. You have all these bundles of collagen fibers, and they run every which way. Bundles of collagen fibers they have no regular orientation. That's why it's an irregular tissue. Have no regular orientation. So on this slide, it's kind of like putting meat through the meat grinder. You have all these bundles of collagen fibers, and when you cut it to make your slide, it kind of appears like this. So we'll look at the dermis of skin in lab as well. Let's move on to, okay. This is elastic tissue. Elastic tissue, just like the name implies, provides elasticity. This is aorta. That's the example I'll give you. So this is from aorta, the largest artery attached to the heart, and we're looking at the vessel wall. So in the vessel wall, you're going to notice primarily two things: pink smooth muscle and black elastic fibers. And they're all mixed together. I mean, look at that big layer of the vessel wall there. I see black and pink all mixed together, so I'll have to be clear. I say, well, what's the black substance? Or what's the pink substance? So the color will be the clue. Black versus pink. Now the function for an aorta is when the heart beats, there's a period of where the heart generates pressure called systole. And then there's a period where the heart relaxes, called diastole. Let me write that down. You'll have to know that eventually. Systole, diastole.
These are related to the systolic and diastolic pressure?
Yeah, actually, and it relates to this vessel. I want you to understand the function of the elastic fibers in the aorta. So when the heart generates pressure, it creates a pressure that's about 120 millimeters of mercury. I'm talking about the heart. Let me write it over here so it's more clear. Talking about the heart for a second, I'll get to the aorta. When the heart generates pressure, it pumps blood. People know that the heart pumps blood, right? You don't have to tell them that. But the top number, when you measure it in the clinic, it generates a pressure of about 120 millimeters of mercury. Millimeters of mercury is the unit of pressure in medicine. When the heart is in diastole and it relaxes, pressure drops almost to zero. The heart is generating no pressure during diastole. It needs to relax and fill with blood for the next pump. But in the blood vessels, you don't want pressure to drop to zero because the blood will stop flowing. What we see in the aorta is that during the systole, yes, you push blood into the aorta and it kind of like expands the vessel wall, and you measure that pressure as a higher pressure. But in the aorta, these elastic fibers provide an elastic recoil during diastole. Provide elastic recoil. During diastole. So that way, there's a pressure drop, but because you pump blood into this vessel and you've stretched out your elastic tissue. But because the heart's in diastole and it's not generating any pressure, during diastole the elastic tissue allows for that little bit of recoil. And that recoil will help push blood forward while the heart's in diastole. So that pressure doesn't drop to zero. Maybe it drops to like 80. So I mean, those are your textbook blood pressure numbers, right? I just want you to know the function of, since we're showing you, why is it there? You need the elastic recoil while the heart's resting for the next pump. Well, anyways, it's not a hard tissue to find if you know what you're looking for. Here's my picture of an aorta. Here's a closer up picture. It projects pretty well. I think what we're going for here is this layer of the vessel wall. Again, the black and pink are all mixed together, so I'll have to be very clear. What's the black tissue? The elastic fibers, or just say elastic tissue. And if I say identify the pink tissue, We are doing smooth muscle in this chapter, so you have to know both. So for the cartilages, remember the name of that cell I want you to know? There's 3 types of cartilages. The chondrocytes, and because the matrix is so dense, it's like they need to push out so they have their own little space to live so they can breathe. That's how I think of it. They're called the lacuna, the space where the chondrocyte is. So that black dot is the nucleus, but you can make out a cell membrane and then even a space around it. So the chondrocytes live in lacuna in their tissue matrix. Let's go over the cartilages. This is hyaline cartilage, I believe. Okay. The best example I like to show students of where it is in the trachea. And then I'll show a picture of the trachea. The trachea is a tube, and like most things, it's soft tissue. But the point of the trachea is to ventilate air in and out. When you suck air through something, the thing could collapse. So the cartilage rings keep the airway open, and this is a hyaline cartilage ring, and they're C-shaped. You surround the trachea, so that when you suck air through it, the stiffness of it keeps the airway open. In the trachea, you have these hyaline cartilage, C-shaped rings. The function, well, in the trachea, the function is to keep the airway patent, keep the airway open. Patent means open. So what you want to look for are the chondrocytes in lacuna. A common mistake is this tissue gets misidentified because you can't recognize the chondrocyte. If you see chondrocyte, you know it's one of the 3 cartilages. Now, the collagen fibers are packed so tightly together, when you stain them, it just has this frosted pink appearance. But they're individual fibers there, you just can't make them out. Fibers packed so tightly. That, along with the high water content, they just look, I'll say frosted pink. However you describe that, I always say frosted pink. Looks like there's no fibers, but they're there, they're just all frosted together. Okay. Well, they also show the costal cartilage as an example. And I think that's good to know since I have Mr. Skeleton here. I think one's a Mr., one's a Mrs. I think this is a female. And so the costal cartilages, they attach they're the attachment for the ribs to the sternum. That's another good example. Because it gives when you do chest compressions on someone, it makes the rib cage a little more compressible. So another example where you find it, costal cartilage. Costal means rib. So the cartilage there makes rib cage more compressible. The cartilage attaches the ribs to the sternum. So those are two good examples of hyaline cartilage. Okay, here's our picture of hyaline cartilage. Yeah, looks okay. So all this tissue here. So I see a cell, that's the chondrocyte, but the little space around the cell, lacuna. Okay. Oh, if I ever have you identify this on anything, just say chondrocyte and lacuna. You get it right. Okay. Don't leave out chondrocyte. Don't leave out lacuna. Just put the whole thing, because they're always, it's a twofer. They come together. All right. Do you remember what color elastic tissue was? Black. So elastic cartilage, we're going to look for this black, dark tissue. And again, look for the chondrocytes and lacuna so you don't call it something else. I don't want you to confuse elastic tissue with elastic cartilage. So again, look for chondrocytes and lacuna. And the black elastic fibers in the matrix makes it very obvious. Ear and epiglottis are the best examples where you find this in terms of location. Ear, epiglottis. The glottis is the airway, so this is a flap of cartilage that guards the airway when you swallow food. Okay, but because it's elastic, well, like the slide says, it maintains the shape of the structure while allowing great flexibility. Okay, it's elastic cartilage. So here's a picture, and in this tissue I see, let me fake quiz you. Identify this tissue, tiny bubbles. Adipose. This is the tissue we're looking at. Even at 100x, I can see the chondrocyte and lacuna, and everything around it is just black. So that's the elastic cartilage all up in here. (400x) looks like this. See the chondrocyte and lacuna a little better, and just the dark black tissue is a dead giveaway for elastic cartilage. Again, not elastic tissue, elastic cartilage. Look for the chondrocyte and lacuna. Okay, I think that's from epiglottis. Okay, this is another cartilage, the third type from the intervertebral disc. It's a very fibrous fibrocartilage. That's what they call it, fibrocartilage. And here you can make out. Can you make out the zigzag fibers? They zig, then they zag, you know, zig. They kind of have this 90-degree angle thing happening. Now this is your spine. And these little discs in between the backbones are cushioning your vertebrae within your spine. So fibrocartilage, its main job is to resist compression. I'm going to leave chondrocytes and lacuna because they're still there. Resists compression. This is one of the major tissue types in the intervertebral discs between your backbones. Found in intervertebral discs. So if you, I've actually done this where I'm dissecting, I get to the spine, and I take a scalpel, and I literally just cut through one of the discs, okay? And I look at it, and I go, ooh, what do I see? When you cut a disc open, it's like a jelly donut. In the middle, there's just this jelly-like fluid, and they call it the nucleus pulposus, some just jelly in the middle. Don't need to know that. What you need to know is the tissue surrounding the nucleus pulposus. They have many layers of fibrocartilage. And so those many layers, they refer to as the annulus fibrosus. That's where you got your cartilage. So this, all these layers of tissue here. Annulus fibrosus. Many layers of fibrocartilage. And what you notice from this picture is, see the outermost layer, maybe they run in this orientation, but then the layer next to it, it runs kind of like at a 90-degree angle. And then the layer into it runs at kind of another 90-degree angle. So this makes it very good at resisting compression, because when the weight of your body coming down on your spine, especially down in the lower lumbar region, these are resisting compression. This is bulging, and these many layers are resisting the compression. And by having orientation, having a different orientation, it makes it better at resisting. Of course, this can rupture, right? We call it a slip disc, or say over time, imagine you have in your chest a big tub of water filled to the very top. Any movement that spills water out is bad for your lower back, because every time I bend forward, I squash my disc, and I make the nucleus pulposus push backwards. And over time, with bad posture, these can tear. And then when this slips out, you get a bulge back there. It can compress nerves and it can cause trouble. Let's see. They say most people, by the time you turn 30, you'll have your first back problem, and that was the case for me. It's not fun. But anyways, I'm gonna turn off the lights. It doesn't stain very dark. Let's notice the zigzags. Do you see it? Oh, okay. Yeah. I'm seeing a layer here, runs this way. This layer looks like that. But I can see the layers, and I can see the zigzag, and I see the chondrocytes in lacuna. So call this fibrocartilage. Zigzag. All right. Moving on from that, those are the three cartilages. This is bone, or osseous tissue. I think as a tissue, it's easy to remember. Oh yeah, that's bone. It's easy to identify. But what are you looking at? Well, there's compact and spongy bone. This is bone, compact bone. Okay. It's spongy layer. Anyways, compact bone. The main structure are these columns of bone called the ostia. And when you look at it in cross-section, it kind of looks, well, you have a central canal, and that's for blood vessels, like arteries and veins, maybe some lymphatics, maybe some nerves. And I'll just draw artery and vein in there. And that canal is surrounded by many layers of lamellar bone. Draw these concentric circles around it. Okay, so there's a couple of layers. Now, in between the layers, you have the bone cells maintaining these layers. They're called the osteocytes. And I'll try to So maybe they're living here. All these little red cells. We'll call them osteocytes. They're stuck between the layers of bone. So all throughout, you're going to have all these osteocytes that live in between the layers. Cells, they have to have nourishment, but we're dealing with a calcified, mineralized tissue. So it's very rigid. So there's all these like little cracks that emanate out from the central canal. So I'll try to like illustrate that. All these like little cracks in the sidewalk kind of looks like. And they emanate out from the middle layer, which is where the blood vessel is, to the outer layers. So it's very important that those little cracks get to all the osteocytes so they can get their nutrition. Okay, and those little cracks are called canaliculi. Central canal, oh yeah, they don't put canaliculi. Okay, let me put that term down. You're gonna have to know that term when we get to that chapter anyways. The little cracks in the sidewalk. Canaliculi. That's how the blood can flow to all the many layers so all the cells get their nourishment. So again, call the central canal, and call the whole thing an osteon. And in this tissue, you see many osteons, but this is the basic structure. A column of bone, that's what an osteon is. The cell type, osteocyte. We'll learn other bone cell types later. So they use the term lamella, referring to the layers. I mentioned that before. Bone, lamellar arrangement. Just many layers. If you want your bone tissue to be strong and have more layers, keep your physical activity level up. Mechanical stress of exercise will help keep your bones strong. So what's this big center? Central canal. And you know, the osteocytes are in a lacuna too. Now this is dry tissue, so it looks like a black space. There's no living cell in there when you prepare this tissue slide. It's just the space where the osteocyte would be. So I guess I should add lacuna. So this would be an osteocyte in a lacuna, but in a tissue sample, the living cell is not there. So osteocytes in lacuna. Blood. I'll keep it easy. There's a whole chapter on blood when you get to 431. You should be able to recognize white blood cells from red blood cells. That's it. So what they do is they just take a drop of blood, they just put it on a slide. They call them blood smears. You stain them. For our blood slide, just be able to tell the RBCs, the red blood cells, from the white blood cells, WBCs. The fancy word is erythrocyte and leukocyte. Erythro means red. Cyte means cell, versus leukocyte. Leuko means white. However, they stain purplish on the slide. And it's easy to tell what's what because the red blood cells have no nucleus. And they're kind of flattened in the middle. So if you look at a red blood cell from the side, they have this, they call it biconcave shape. Biconcave means curved, curves in. So biconcave means it's curved on both sides, right? It's curved there, and it's curved there. So when you look at this through the microscope, the light shines more through the center than the sides. That's why it appears more translucent in the middle. And there's no nucleus. So, and it's like they outnumber the white blood cells like 1,000 to 1. So it's mostly red blood cells. You might see a couple of white blood cells. So red blood cell, white blood cell. This is a lymphocyte. This is a neutrophil. There are types of white blood cells. For this first test, you just call them white blood cells. They're the ones with the fancy colors. They're larger and they have these weird nuclei. This one's a large round nuke with a little rim of cytoplasm. This nucleus has many lobes. And I don't know why it has that pinched appearance, but it's a neutrophil. Right, that's all I'll ask you to do with the blood. Here's a blood slide. Turn the lights off. Which cell am I pointing to in those? Those are the white blood cells in a sea of red blood cells. Okay. Again, those are white blood cells. Most of it is red blood cells. So just be able to tell red from white. There's another picture. Both of those are white blood cell types. And you'll be able to identify the different ones and know their functions when you study that chapter in 431. Just know this as blood tissue for now. So I just made a list of all the connective tissues I've covered. And when you take note of that, take a picture of that. Before class, I put the locations down. Why don't I do this? Why don't I copy that to here? So when I go through the slides, you'll know where I am.
function.